Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
PYR Test. Principle, Media, Procedure, Results, Uses. Table of contents is Introduction, Objectives, Principle of PYR Test, Requirements for PYR Test, Procedure of PYR Test, Result and Interpretation of PYR Test, Quality Control, Precautions, Applications of PYR Test, Limitations of PYR Test. Introduction. The PYR test, L-pyrrolidinyl B-naphthylamide test, is a biochemical test used to detect the ability of bacteria to produce the enzyme pyrrolidinyl aminopeptidase using L-pyrrolidinyl B-naphthylamide, PYR, as a substrate. It was first reported in 1981 by Godzi J., Schulman R., and Eric L.A., who used PYR as a substrate to identify Streptococcus pyogenes and Enterococci. This test is now widely used in clinical and research laboratories for diagnostic purposes. It is primarily used for the detection of Streptococcus pyogenes, Enterococcus spp, E. coli, and for the identification of Staphylococcus spp. There are two methods currently in use for performing the PYR test: the tube method, which uses PYR broth; the disc method, which uses a PYR impregnated disc. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Objectives. Assess the ability of bacteria to produce the enzyme pyrrolidinyl aminopeptidase. Biochemically differentiate and presumptively identify sample bacteria using this assessment. Principle of PYR test. If bacteria can synthesize the pyrrolidinyl aminopeptidase enzyme, the L-pyrrolidinyl B-naphthylamide substrate in the broth or disc is hydrolyzed. This hydrolysis releases B-naphthylamide. The released B-naphthylamide reacts with N-dimethylaminosinamaldehyde. This reaction forms a bright pink or cherry red Schiff base. The appearance of this color indicates a positive PYR test. Requirements for PYR test. A. Culture media. PYR broth is used for the broth, tube method of the PYR test. Composition of PYR broth per 1,000 ml. Beef heart infusion, 500.00 grams. Peptic digest of animal tissue, 20.00 grams. Dextrose, 2.00 grams. Disodium phosphate, 0.40 grams. Sodium chloride, 2.00 grams. Sodium carbonate, 2.50 grams. Chromogenic mixture, 0.10 grams. Final pH, 7.8 ACE 0.2 at 25 degrees Celsius. Alternatively, PYR agar medium can be used. PYR agar will have all the components of PYR broth with the addition of 15.0 grams of agar powder to make it solidified. Preparation of PYR broth. Measure the appropriate amount of PYR broth, agar, powder, or the media components. Mix in the required volume of water in a conical flask or glass bottle according to the instructions of the manufacturing company. Stir well using a magnetic stirrer or manually and heat to boiling to ensure all components dissolve completely in water. Dispense about 5 ml, or the required volume, of the medium into test tubes. Tighten the caps of the test tubes or cotton plug them. Autoclave the tubes at 121 degrees C and 15 pounds pressure for 15 minutes. Let the tubes cool to around room temperature, below 40 degrees C. B. Reagents. PYR reagent use 0.01% PN N-dimethylaminosinamaldehyde, or 1% PN N-dimethylaminosinamaldehyde, as the PYR reagent. Composition of PYR reagent: N-dimethylaminosinamaldehyde, 1.0 grams. Concentrated hydrochloric acid, HCl, 1.0 ml. Distilled water, 99.0 ml. Preparation of PYR reagent. Add 1.0 gram of N-dimethylaminocinnamaldehyde to 99.0 ml of distilled water. Slowly add 1.0 ml of concentrated HCl with constant stirring. Mix thoroughly until the N-dimethylaminocinnamaldehyde is completely dissolved. Store the solution at 2 to 8 degrees C in a dark place. PYR impregnated disc for the rapid method or disc test. C. Equipment: test tubes, forceps, weighing machine, autoclave, Bunsen burner, incubator, inoculating loop, Petri plate, PPE and other general laboratory materials. D. Test organisms, sample bacteria. Positive control: Streptococcus pyogenes, ATCC 19,615 inches. Negative control: E. coli, ATCC 25922. Procedure of PYR test. Tube method: PYR broth or agar method. Using a sterile inoculating loop, pick sample bacteria from a well-isolated colony of fresh culture, 18 to 24 hours old. Inoculate the PYR broth with the bacteria. Alternatively, prepare a 0.5 McFarland standard suspension of the sample bacteria and transfer a loopful of the suspension to the PYR broth. Incubate aerobically at 35 degrees C for about 4 hours, or 18 to 24 hours for PYR agar. Add 2 drops of PYR reagent to each tube. Observe for a color change within 1 to 2 minutes after adding the reagent. Rapid method: PYR disc method. Place a PYR disc in a sterile Petri plate using forceps. Moisten the disc with sterile distilled water. Using a sterile inoculating loop, pick up 1 or 2 loops full of well-isolated sample bacteria, preferably from blood agar plate, hemolytic colonies from BAP, and rub them onto the PYR disc. Transfer several loops full of bacteria if the sample bacteria is slow-growing and requires 48 hours or more of incubation for growth. Incubate the plate aerobically at room temperature for 2 minutes. Extend the incubation to about 10 minutes for slow-growing bacteria. Add 1 to 2 drops of PYR reagent directly onto the PYR disc. Observe for a color change within 1 to 2 minutes after adding the reagent. Result and interpretation of PYR test. A bright pink or cherry red color developing over the disc or in the broth, agar, within 1 to 2 minutes after adding the PYR reagent indicates a positive reaction, PYR positive. No color change or the formation of blue, yellow, or orange colors after adding the PYR reagent indicates a negative reaction, PYR negative. The development of a slight or faint pink color also indicates a negative reaction. PYR positive bacteria: Streptococcus pyogenes, Enterococcus faecalis, Enterococcus faecium, Citrobacter species, Klebsiella species, Yersinia species, Staphylococcus hemolyticus. PYR negative bacteria: E. coli, Streptococcus agalactiae, Streptococcus bovis, Salmonella species, Staphylococcus aureus. Quality control. Streptococcus pyogenes, ATCC 19615, results in the formation of a bright pink or red color on the broth, or over the disc agar, within 1-2 minutes of adding the PYR reagent. E. coli ATCC 25922. Does not change color, remains unchanged after the addition of the PYR reagent. Precautions. Store the PYR reagent in cool, dark conditions. Moisten the PYR disc slightly. Do not saturate it with water. Ensure to transfer a sufficient inoculum and rub it thoroughly over the disc. Using too little inoculum might lead to false negative results, characterized by the formation of a faint pink color. Do not interpret faint pink color as indicating a positive reaction. Applications of PYR test. To distinguish Streptococcus pyogenes from other B. hemolytic Streptococci. To differentiate Group A and Group D Streptococci, PYR positive, from other Streptococci. for identifying E. coli among other gram-negative bacilli that are indole positive and lactose positive, to distinguish Citrobacter spp, PYR positive, from other H2S positive gram-negative rods, such as Salmonella spp, PYR negative. Limitations of PYR test. Additional biochemical tests are necessary to confirm the complete identification of bacteria. Excess moisture on the disc can lead to false negative results. Taking inocula from selective or tube biochemical test media can result in false negative results. Using inadequate inoculum may lead to false negative results. Results should not be interpreted before 1 minute to avoid non-specific outcomes. E. coli and other indole-positive bacteria may exhibit a blue color on the media and disc, which is considered PYR negative and can be confusing. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!